Right. Good afternoon, everybody. Welcome to the SACS Forum Cell and Gene Therapy. I have an illustrious panel today of experts that represent our field very broadly across a big spectrum of novel technologies. And I hope that we can give you some important insights today as part of this panel discussion. So the way we'll go is we'll do quick introductions, set the stage, and then we'll go into moderated questions and answers. So for all of you to follow. Let's begin with introductions. My name is Axel. I'm the head of oncology at GSK, and part of the GSK portfolio is cell and gene therapy, particularly in oncology, which is the focus for me on today's panel. So let's go around the table. We have Ben next.
Excellent. So my name is Ben Burnett. I'm a sell-side equity research analyst for Stifel. I cover primarily stocks of companies developing oncology and cell therapy-based medicines. Excited to be a part of this panel. There's an incredible breadth of innovation represented here. And I just wanted to say, from a financial perspective, I think I'm the one person up here who's not an innovator, looking at this sector from a completely financial perspective. I just want to say off the bat that I think this sector holds certainly risk, but a lot of promise as well. There's a ton of precedent to draw from bone marrow transplantation, which is a cell therapy at the end of the day. But there's a long history of curing patients with certain blood cancers with transplantation. CAR T, of course, is more recent. Enormous amount of ground that still remains to be uncovered. But I see this category as one that will continue to attract capital. And in certain opportunistic situations, I expect we'll continue to see major de-risking events happen relatively early, phase 1-2, rather than phase 3. And when that happens, we start to see valuations really price in quite a bit of optimism early on. So an exciting space. And again, happy to be co-moderating this panel. There's a lot of interesting but unique, diverse approaches represented here. So I'll stop talking and hand it over to, I think Daniel, I believe you're next. Is that right?
That's right.
So I'm Daniel Teper. I'm the principal founder and chief executive officer of Cytovia Therapeutics, and we focus on natural killer cells.
That's great. Jing, you're next.
Thank you. Hi, everyone. Great to see everyone today. My name is Jing Zhao. I am the chief business officer and the CFO of Refuge Biotechnologies. And we are playing in the field of programming cell therapies to develop much more powerful cell therapies to bridge that gap between hematological oncologies and solid tumors.
Excellent. Thank you. Michael.
I'm Michael Klichinsky. I'm one of the co-founders and the senior vice president of research at Carisma Therapeutics. We're a clinical stage cell therapy company developing CAR macrophages for solid tumors.
Excellent. All right. Thank you so much. Sid?
Yes, thank you, Axel and SACS Associates for inviting me here. My name is Sid Kerkar. I oversee research and development for EXUMA Biotech. We're focused on developing next generation CAR T therapies. We're excited to be here.
Great, thank you. Mythili.
Hi, I'm Mythili Koneru. Call me Mai, and I'm CMO of Marker Therapeutics. We work on non-engineered multi-tumor associated T-cell specific therapies. I'm very happy to be part of this panel. It's quite diverse. I probably am a minority in the sense that I work on non-engineered T-cell therapies, and I'm excited to discuss these topics in more detail.
All right. Did I miss anyone? Then we're good. So then let me set the stage for this conversation by just saying a few words about the cell and gene therapy field. And we do have a few slides. So some of our presenters will just show a slide or two as part of their introductory comments. So if you could start with the first slide, please. And there's the first slide. Okay. So, the cell and gene therapy field has dramatically evolved over the last several years since the first CAR T approved about a decade ago. So it's an exciting area, but it is also one of the most complex areas in medicine. And as we see the next generation technologies emerge and make big contributions to the space, we also need evolution in methodology, more sophisticated and eventually, systematic thought process around how to develop them and where to develop them. And then eventually improving the patient journey for cell therapies to be more readily available for patients, and reducing some of the challenges that we still see in the supply chain. So I say a few words about those individually before we go to the panelists. So if you look at this slide, the information here comes from a recent review about cell and gene therapy and the industrialization, so solving those challenges I just named. There are three things on here that I'd like you to be aware of. The first one goes to the cell therapy utility framework, which is basically an attempt to systematically categorize which diseases, and this is focused on oncology, which diseases would benefit more versus less from a tailored cell and gene therapy approach. And it's focused on two key components: the ability of a tumor to develop a tumor microenvironment that basically protects from immune destruction. So an tumor microenvironment that is protective, and then the ability to hide from the immune system by reducing antigen presentation or low tumor mutation burden or any other feature that reduces the ability of the immune system to see the tumor. So if you take those two things together, you can create four major categories of tumor types, of which some lend themselves very well to cell and gene therapy and others don't. So I encourage you, have a look at this. This could help focus the development of a cell therapy in oncology on the tumor types where particular value can be derived. Now, in order to do this well, we also need to increase the efficiencies. Efficiencies can come from new clinical methodologies, such as the recent FDA introduction of a parent-child IND that allows you to investigate multiple variations of the same cell and gene therapy under one umbrella. And one can do that in a master protocol where the first generation of a cell therapy, the first that reaches the patient, can be improved either through a CMC process or through further genetic engineering. And we'll hear more about that in a minute through the panel. And then you can test them side by side in one trial rather than do iterative trials that take a substantially higher amount of resources and time. So the parent-child IND could be a significant improvement in efficiency for clinical investigation. If you go to the next slide, other challenges that patients and developers still face are within the patient journey. And as we all know, we have to, so far with autologous products, we have to remove cells from the patient, we engineer them, and then they go through a complex CMC process and supply chain process before they reach the patient again to be reinfused. Many steps along this way, from developing the companion diagnostic that identifies the right patient for the right product, the apheresis, several steps on the CMC side, and the lymphodepletion that is quite burdensome for patients before the cells can be reinfused. All of that has opportunities for improvement and would help what I simplify as industrialization of the process, making it simple enough that you can use it like a regular medicine. We're not there yet, but I think all the components and the innovation that each of the panelists are bringing to the table can help making that supply chain more patient-friendly and increase, of course, the activity of the compounds that ultimately drive adoption. So it's an exciting time to see all these things come together, even though we're in the middle of this journey. We're not there yet. So I'd like to, take this as just the platform from which we will have the conversation today, and now go to our panelists and ask them the question that each one of you should be able to answer. Think, at the present state of affairs of cell and gene therapy, is a particularly exciting area of innovation. What do you think is an exciting area you want to say a few words about that all play together into this new space? And we'll start again. So Ben, it's your turn.
Oh, well, I think I'll move that question to maybe I'll start with Daniel, first innovator. Go ahead, Daniel.
All right, that's fine.
Sure. So thanks. Axel, you talked about industrialization of cell therapy, and we believe that autologous T cells have been a real breakthrough in that they provided cure to patients that had no option left. The question is, how can you bring cell therapy to more patients and earlier in the treatment? The approach that Cytovia Therapeutics is taking is to focus on natural killer cells, which are allogenic, NK cells, allogenic and off the shelf and naturally. So where do we differentiate ourselves, and where do we think that we bring value? We actually the first company that is combining two modalities toward NK cells. The first one is the development of gene-edited NK cells from induced pluripotent stem cells. Has significant advantages in terms of industrial scale-up, low cost of goods, one-time editing and reproducibility. And the second platform that we think is complementary are NK engager bispecific antibodies. And we've seen recently at AACR that combining NK engager antibodies with cell therapy could be a winning strategy and could be administered on an outpatient basis earlier in the treatment in multiple diseases, and not just in hematological malignancies, but also in solid tumors. So we see a great future for cell therapy overall, and we're looking for solutions to bring cell therapy to more patients.
All right, Daniel, thank you so much. I would ask the same question to Jing.
Great, thank you. Fantastic, thank you for putting the slide up. So I think we were one of the earlier adopters of looking at how can you have a cell react in its own way to the cellular environment, rather than depending on external forces or other ways of sort of manipulating the cell. So, because the idea is, if the cell can actually clear tumors very originally, then how can you elicit that response back? And we were one of the first people to take more of a synthetic biology approach to this. So I think it's actually been very exciting to see that as this has developed over the last few years, there are now many more companies coming to the fore with different ways of, as we call it, sort of cellular programming, in its ability to program responses for these cells so that once they come into contact with tumor antigens, or once they're in a particular environment, they do a specific thing. At Refuge, we have a platform where the idea is we can link together any type of sensing receptor or reactionary receptor on a cell and have that directly go into the nucleus with a genetic response. So we modulate, as opposed to edit the genome, so that there is a up or down regulation of specific genes so that the cell can become more powerful or become more durable as we now understand cell therapy is better to get better responses in patients. And for us, the idea here is, we're relatively agnostic to saying, there's only one type of cell that works. So, we're able to apply this technology, this synthetic biology approach to any type of cells, including T cells, TILs, macrophages, NK cells, etc. Internally, we play with CAR receptors and T cells to date to demonstrate the power of this approach. We just announced the collaboration yesterday morning with the MD Anderson on applying our technology in TILs. And within this field of programming, as we see more players coming to the fore, rather than competition, I think it's nice because then we can actually learn more off each other. And we're looking forward to more of these new modalities being in the clinic and then ultimately being in the market.
Yeah, I'm giving the word to Ben to drive the rest of the questions.
Okay, well, let's just, with that same theme, let's move on to the next panelist, which I think is Michael.
Hi, everyone, and please let me know if my audio freezes again. As I mentioned, I'm Michael Klichinsky from Carisma Therapeutics, and Carisma is focused on a novel cell type, the macrophage, and applying genetic engineering of macrophages towards solid tumors. As we all know, CAR T cells have been tremendously effective in blood cancers with CD19 targeted CAR T cells, leading to multiple approvals and tremendous responses, transformational responses. But in solid tumors, regardless of the target antigen or the type of cancer or the type of T cell engineering approach, et cetera, we're seeing limited responses. And there's three general barriers, or three buckets of barriers. The first is trafficking. The second is the immunosuppressive tumor microenvironment. And the third is target antigen heterogeneity. And we believe that by using myeloid cells, macrophages, and monocytes, we have the potential to overcome these key barriers. Firstly, as many of you know, macrophages are the most abundant immune cell in solid tumors, which speaks to the fact that tumors go out of their way to recruit these cells. The chemokine receptors, the integrins, the enzymes and proteases that allow these cells to not only home toward tumors, but extravasate out of tumor neovasculature and penetrate tumor tissue are distinct and unique from other immune cells. We found that by putting CARs into macrophages, we can directly tell these cells what to eat, and they will phagocytose target expressing cells. And through the process of phagocytosis, they will kill the target cells, directly debulk tumors. But critically, as macrophages, we can also polarize them to an M1 pro-inflammatory phenotype, and not only direct them to kill, but direct them to reprogram the TME. And we find that these CAR macrophages directly are recruiting in other immune cells, T cells, NK cells, dendritic cells, B cells, et cetera. They induce MHC expression on the tumor cells. They produce Th1 skewing cytokines and induce a favorable environment for a robust immune response. And critically, macrophages are one of the professional antigen presenting cells of the human immune system, along with dendritic cells and B cells. And we find that our CAR macrophages will directly present and cross-present tumor-derived antigens after CAR-mediated phagocytosis, leading to epitope spreading, a broad adaptive immune response, and critically, protection from antigen-negative relapse. And as you know, antigen heterogeneity is a key challenge in the solid tumor setting. So being able to directly recruit other arms of the immune system and lead to a broad innate and adaptive immune response, we think is critical toward making a dent in solid tumors. Carisma is currently a clinical stage company. Our lead program is targeting HER2, and we announced about a month ago that we treated the first patient with a CAR macrophage. It was the first patient to receive an engineered macrophage, and that study is actively ongoing and recruiting. And I'll stop there. Thank you.
Fantastic. Mai from Marker Therapeutics, over to you.
Thanks. I appreciate it. So really to address Axel's question about what exciting development in cell therapy, my take on it would really be this minimal engineered or non-engineered approaches, and that's being obviously advanced much further in solid tumors. I think these approaches, were a little bit later to the party compared to obviously CD19 CAR T cells in heme malignancies, but I think they're making great advances in the solid tumor space and improving their ability there. Obviously, we just heard Maria talk about at Iovance about the TIL therapies and the efficacy that's being seen in various tumor types, including melanoma, cervical cancer. And I think really what's appealing about it in solid tumors in particular is the broad antigen profile. And that's really the approach Marker is taking as well. So at Marker, we deal with a heterogeneous tumor by selective expansion of the non-engineered tumor-specific T cells. And they recognize tumor-associated antigens. And so we refer to them as multi-TA T cells. So the idea here is that by targeting 4 or 5 different antigens, you're getting much broader spectrum of anti-tumor activity. So the idea is that with these cells, there's a lot of unique features to it compared to other cellular therapies. You actually don't require any lymphodepletion. And so by not needing lymphodepletion, you're actually able to recruit the endogenous immune system. And this can actually lead to this phenomenon of epitope spreading. And we feel that this ultimately contributes to much more durable responses. Because not only are you getting the initial expansion of the infused cells, but you're actually able to induce a secondary immune response from the patient's own immune system. So the idea here is that marker really targets antigens via the native T cell receptor. And by not needing genetic engineering, you're actually reducing the cost of production ultimately. And you're actually able to recognize both extra and intracellular targets, thereby broadening your scope. And in addition, they're actually quite easy to administer. It's actually a 10-minute infusion in the outpatient clinic setting, so you don't need an ICU or hospitalization stay. And in over 150 patients that were treated, there was no evidence of cytokine release syndrome or neurotoxicity. So the administration is not only easy, but the toxicity profile is much more favorable than, for example, CD19 CAR T cells. And this profile allows it to be administered not only in academic institutions, but in a community setting, as well as in the adjuvant population. So patients, for example, that don't have active disease, but are in remission, but maybe high risk of relapse, you can actually administer these cells. The antigens that we target are in our autologous product are PRAME, MAGE4, SS62, Survivin, and ERPSO1. And these particular antigens were chosen because they're cancer testis antigens. So they're much more broadly expressed across a variety of different tumor types. And so the idea is that you isolate apheresis material from the patient, and then you take the dendritic cells, you pulse them with the peptides. So we have a library of peptides that span all 5 of these antigens. And so you use those peptide pulsed dendritic cells to stimulate the T cells. And so what you have is a product that are T cells that are very specific for those antigens of interest, and then the non-specific cells sort of die off. And then the cells are tested for purity, potency, et cetera, and then they're later infused into the patient. So I think this approach of just, dealing with the heterogeneity that's inherent in a lot of patients is by just, broadening the antigen specificity of the product ultimately. Thank you.
Fantastic. Sid, how do you think about this at EXUMA?
Yes, at EXUMA, we're really working on some exciting developments, and we're looking to solve 3 major problems that we believe are the largest hurdles for cell therapies and gene therapies. One is really the limited success to date for solid tumors. There's been a lack of efficacy, and we've all discussed this on the panel. Our approach is to go with what we call a logic-gated CAR T, which is applicable to validated tumor targets. And it's developing a CAR T that recognizes the target very specifically in the tumor microenvironment due to the Warburg effect and the lower pH in the tumor microenvironment. And we believe that this is an approach that can work for a lot of different common targets across 90% of all solid tumors. And this is a program that is in phase 1 trials. But secondly, we're addressing the complexity of delivery and safety issues. We know that one of the issues is that CRS and neurotoxicity. We're really looking at this through multiple avenues. In one particular way we want to do is give a subcutaneous T cell CAR T therapy without lymphodepletion. For multiple reasons, we feel that biologically this will be a safer, more kinder, gentler CAR T that can be rapidly generated and given in a chemo-free environment. And we know that the kinetics will be different in given subcutaneously versus intravenously. And this is a program that we have very good preclinical data for and that we're looking to take to clinic next year. And along with the subcutaneous delivery, we've built a system where we can manufacture these cells the same day. And we've really spent a lot of our discovery efforts on synthetic biology in getting our lentiviral vectors to a point where we can target specific cell populations and immune cell populations by viral engineering. And then as we have synthetic elements in the actual viral vector that can make these cells expand, proliferate in a lymphoreplete environment, and also really be able to resist the negative factors of a solid tumor microenvironment. So these are the problems that we feel are the most important to address, and are excited to see these programs develop.
Fantastic. Okay, obviously lots of different approaches, and I think everyone kind of touched on this a little bit within their within their discussions. But I want to ask one question, and then I'll hand it over to Axel. Addressing the challenges of the tumor microenvironment, how do you think about those challenges and getting around that? And also, kind of what comes with that is addressing antigen escape. Those challenges have been tough. How does your approach enable you to win there? And we can go around. We can go around the panel as we did before. And if you want to, Dan, do you want to start or?
Sure. No, I'll start. There are different ways of approaching the tumor microenvironment, and obviously different cells are proliferate more in certain organs. So, one organ of interest to us is the liver, because we have a GPC3 program for hepatocellular carcinoma, and it's been demonstrated that NK cells are very active in the liver against HEC cells. I mean, obviously the challenge is that as the disease is progressing, the NK cells are exhausted and need to be replaced by new cells. So the question is, how do we provide those cells? And we derive NK cells from iPSC, which allows us to get off the shelf and cost effectively large quantities. But that in itself, may not be sufficient. So then we go to gene editing. And gene editing, the challenge is that can you get consistent product, and do you have to do editing on every batch? So our approach is to do the gene editing, and we announced a few months ago a partnership with Selectus, and talent gene editing is to do gene editing on the stem cells, and you do it once, and you have a master cell bank of edited iPSC, which then you differentiate, expand, and cryopreserve, and it's available to many patients. So that's a cost-effective way of doing editing. Question is what you edit, and then there's different approaches. Our initial focus is on TGF-beta2 receptor knockout, which is well known in the regulation of the tumor microenvironment, but there's a number of other edits that could be done. You may want to do multiple edits, and if you can do those multiple edits at once, on the stem cells, obviously you are reducing the cost of doing that. And then last but not least, in the editing, if you can identify the optimal loci to do the editing, you may improve significantly precision and persistence of your cells. So that's the approach in cell therapy. The question is the cell sufficient, or what's the benefit of combining with monoclonal antibodies or bispecific antibodies? So a number of experiments, including clinically, have been done with monoclonal antibodies, but that's more in hematology. We believe that bispecific antibodies, NK engagers, are particularly attractive, and we are using NKp46 as an engager, and NKp46 is overexpressed, including on infiltrating NK cells in solid tumors. So by combining the two, you can really address different clinical situations, including earlier. And I think, as was mentioned by several of you, if you can move from administering cell therapy only in major academic major centers to a broader number of hospitals, and potentially to more even in the community setting, then, and you can do that at the lower cost, basically at the cost of a biologic product, then I think you will provide the benefit of cell therapy to many more patients.
Excellent. Okay. Maya, what are your thoughts on this?
Yeah, I'd love to try to address some of these issues. I mean, I think to begin with, it's very different, for example, in solid tumors and in heme malignancies. The issues really, with solid tumors is dealing with that tumor microenvironment and antigen loss. Obviously, you don't have that same issue of tumor microenvironment in heme malignancies because the cancer cells are circulating. So if you think about these two issues, particularly in the context of solid tumors, it's really finding the right antigen targets. And then once the cells are able to find the target and enter into the tumor, as Michael said, was a couple of the issues. Then trying to survive that toxic, TME is important. So in terms of finding the right antigen, I think the issue is really about diversification. You need to deal with the heterogeneity that's inherent not only between patients, but even within a patient. For example, at different sites of metastasis, for example, or even as the tumor changes over time, that the tumor is not stagnant, force. And so that's some of the issues, for example, with the CD19 CAR, where there may be, for example, loss of that antigen or lower expression. So the only way to really deal with that is to have a heterogeneous product or a product that can evolve with a changing tumor, whether that's, the ability to change the tumor microenvironment to create a more pro-inflammatory response, so that you can actually recruit, for example, the endogenous immune system is another way to sort of deal with that. So with that, switching over to the tumor microenvironment, as I mentioned, you really have to change that sort of milieu into a place that the immune system can thrive. So, one approach some companies have taken are armoring the CAR T cells. When I was at Sloan Kettering, we worked on, for example, an ovarian cancer or the MUC16 CAR, armoring them with IL-12. So not to say that necessarily is the answer, but just somehow overcoming that sort of blockade of dealing with that very anti-inflammatory environment to try to tip the scales a little bit. The other approach might be combinations, for example. I think typical combinations that a lot of cell therapy approaches have taken are combinations with PD-1 agents, or even able to T cells that can actually even secrete PD-1 has been approached. But I think some of the cell therapies are limited by the toxicity. You can't really combine two agents when one of them is extremely toxic. So maybe even sequencing is going to be important moving forward. I think with Marker, our approach with pancreatic cancer is to combine in frontline metastatic pancreatic cancer with chemotherapy, which we've done with some interesting efficacy results. So I think those are just some ways to kind of deal with both the antigen loss and the TME issues.
Okay, that's great. Michael, anything to add there?
Yeah, I'll just focus on the antigen loss side. I think it's a really critical challenge. And we know that with CD19 targeted CAR T cells, where patients start off with 99.99% CD19 overexpressing target cells, you still get a substantial fraction, something like 30% of patients coming back with CD19 negative relapse after treatment with those CAR T cell products. In solid tumors, the risk is significantly greater, where you start with a lower fraction of your cells expressing your target antigen. So being able to induce epitope spreading and instilling activity against patient-specific secondary tumor antigens, I think, is critical. And we've conducted some experiments specifically looking at the ability of CAR macrophages to have this effect. And what we did was we grew HER2 overexpressing tumors and treated these animals with CAR macrophages, and took the animals that had complete responses and re-challenged them down the line with tumors that were edited to remove the HER2 targets. They were completely HER2 negative. It's a model of antigen negative relapse. And we found that all the animals that previously were cured by CAR macrophages were protected from antigen negative relapse. They had T cell immunity that recognized secondary neoantigens driven both by CD8 and CD4 T cells. And we think this is a critical point to be able to drive endogenous immunity. The second point I'd like to make is one thing we've been looking at are tumor models that are resistant to PD-1. For example, the CT26 colorectal cancer model is well known to be PD-1 resistant. And we found that by using CAR macrophages, which recruit T cells and induce MHC expression, we've been able to drive responses to PD-1 in tumors that are otherwise PD-1 resistant. We think this is quite a notable finding that we're instilling this long-term immune memory and turning T cell checkpoint inhibitors into more effective therapies for otherwise resistant models. And I think it's a critical point to make is that these cells all work together, macrophages, T cells, NK cells, and others. And being able to engage them all is quite critical. And in fact, one thing we're beginning to look at now is the combination of CAR macrophages with CAR T cells in a collaboration with Carl June at the University of Pennsylvania to see if CAR macrophages can in fact help the CAR T cells get there to the solid tumors and evaluate synergy.
Okay. Well, very good. I think that's pretty comprehensive. Axel, you want to kick off the next question?
Sorry, I just have to unmute. It's a little tricky here with the system I have. So thank you for that. A simple comment I would like to make before I move over to the next question, which is basically, given the complexity of what you just heard, how do we actually address, resistance or make these cell therapies most efficient against complex tumors with, variable tumor microenvironments and, therefore useful for as many patients as possible? We need the ability to test multiple things side by side, different engineering steps. And, I like the terminology of, cellular programming, as Jing has been introducing it. I also like the terminology of second generation or next generation engineering, which in my mind is actually quite the same thing. So if you find a way to take all these engineering steps and make them modular, so you can puzzle them together, you have multiple modules of changes to the cell that optimize the cell's effectiveness in a given tumor microenvironment. And there is a lot of variability, so you might have to puzzle these things together in different ways. Then you actually will be able to effectively test them for the right patients. So the modularity of taking all these technological approaches and bring them together will probably give us a chance to find the right approach for the right patients. So that's just a technical addition to all the science that you guys have introduced. So, and I'll stop here on this topic, and let's go to something different, just to increase a bit the diversity of the questions here. Go to CMC for a moment. Obviously, in cell therapy, the product is very closely associated with the process. Some people say the process is the product. So if we say, CMC is that important, what do we think are the next CMC improvements we need to make to optimize products for, clinical use? Start with clinical trials, but then go also to, potentially marketing in a cell product. So, and one of the key features is vein-to-vein time. So how quickly can we actually turn around a cell therapy before, it's too late for a patient to wait for receiving it? So, what are the features that should drive that on the CMC side? That's the next question. Who would like to begin taking that question?
I can start. Obviously, believe that autologous has a role to play, but there's a significant advantage if you can industrialize, allogeneic. And then the question was posed of programmable cells, which I think conceptually is very elegant. But in order to prove that in patients you're going to have the right programming, I think it's going to be challenging, and it's probably years away. The question is, whether you have a manufacturing process that's plug and play. And that's the reason why we prioritize iPSCs, because you can very easily do multiple edits and multiple series of edits. And that's done relatively quickly. You have a master cell bank of edited cells, and then you can produce them also quickly and consistently into NK cells, edited NK cells or CAR NK cells. And you can try them in patients, and you have a consistent product. And whichever is the highest performing product can be taken further into development. I mean, at this point in time, nobody is going into hundreds of patients early on in cell therapy. I mean, everybody is happy to have data on 4 patients, 10 patients, sometimes even 1 or 2 patients. So if you can produce multiple products consistently and cost effectively, I think we'll find which one is the highest performing and can move forward into development. So yes, manufacturing is important, and right now very few CDMO have experience. So I think that the companies that have the most potential, at least in the next several years, are the companies that master the manufacturing and can do it in-house and provide off-the-shelf products to clinicians.
Why don't we just, go ahead.
If I'll keep it short. I think, the CMC improvements, a lot of it, I think, is centered around cost today of autologous manufacturing. And there's probably no right answer to this, but given, Axel, as you mentioned, the complexity of tumors and cancer as a disease, right? It's rarely very homogeneous between 2 different patients. And so, that inherently to me sometimes says, autologous obviously is very individual dependent, right? You're taking an individual cells and giving it back to them. But skipping that back to more, can you really give one complete set to multiple patients? I'm wondering if there's a little bit of an inherent conflict in there. And in any case, I think, in the short term, with the gap that we have in terms of getting things working in solid tumors to address the bigger market for just all the other cancer patients out there, it's probably going to happen in autologous first. And then therefore, so what are the things that, can reduce the cost there for manufacturing of just autologous therapy in the first place? I think one of the key parts of our collaboration that we just established with the MD Anderson will be looking at addressing that, including standing to what you mentioned on, putting together closed systems where, things go in one end and it comes out the other end, it's all standardized, and then just having that replicated in a bigger form so that you can somehow then, get much more economies of scale for even autologous therapy today, without necessarily having to say, okay, well, we have to fundamentally change, into other allogeneic approaches and all of these things where we're effectively solving for GVHD and all those good things. Sorry, a couple of 2 thoughts.
Okay, so one more speaker to this, and then we'll move. So Sid, you're next.
Sure. CMC is always a complex issue. And certainly when we've built a technology where we can generate a product the same day, and the biology is there, but the regulatory science and the CMC science also have to be built. And so to answer your question, Axel, it's really about investing in the science of CMC, in the science of understanding what regulators want. One of the big things is potency, and also to be able to have release criteria for your cell product. And so to be able to do that during development and moving programs from research to development and developing a strategy and developing that dialogue with regulatory agencies to come up with a way and a path to IND is a critical step in programs. And it's certainly a path we're taking with a very unique process where we can actually use a lentivirus and gene modify cells in 4 hours. If you had asked me 10 years ago, I would have said that's not possible. You need to activate T cells to be able to put lentivirus and a gene into a T cell. But we now can do it in 4 hours through engineering the virus, through having moieties on the cell surface and the envelope of the virus. So technologies are much more advanced. And now we have to work with regulators to be able to define novel ways of looking at meeting the specifications that are necessary.
Yes, and CMC will always be at the forefront of, driving success for cell therapies. That will be true. And potency is a critical feature as much as vein-to-vein time. So thank you so much for that. Then let me ask the extension of this CMC question, going from autologous products, which we all know are the centerpiece of cell therapy today, but there is already the notion that allogeneic products might actually be the next generation, might replace them. I personally am not so sure if that's going to be the reality that we're going to see. But, these are opinions. There's no clarity on the exact future yet. So I'm curious to see how you see the allogeneic future of cell therapy, the perceived allogeneic future, or if there are even other alternatives to that. So who would like to begin with that?
Maybe
Sure.
I can start.
Yeah, Michael, why don't you take this one next?
So our approach is to focus on autologous, but we see allogeneic as essentially, and this is the term you used, Axel, an evolution of the platform. So our goal is to focus on autologous to generate proof of concept to understand the biology, and moving into an allogeneic CAR macrophage product is certainly feasible. Macrophages do not cause graft versus host disease. They are less likely to be detected by NK cells for a number of reasons, and the potential is there. But I think it's important to focus on autologous for an unproven platform to generate that core data and enable an approach toward allogeneic. So our current ongoing clinical trial is with autologous patient-derived CAR macrophages. We have established a collaboration with the Blazer Lab at the University of Minnesota to make iPSC-derived CAR macrophages, and are working on optimizing that process. So the potential is there, but I think it's important not to jump to allogeneic until you really know what you have with real autologous cells. The other comment I wanted to make is there are ways to drive down cost and variability with an autologous process as well. And the way that we are approaching that at Carisma is we have a unique opportunity in that macrophages do not need to be expanded, and they don't need to be activated to be transduced, like with, for example, T cells with lentivirus. And we've established a process of engineering monocytes without differentiating them ex vivo, and directly putting those monocytes back where they turn into macrophages within the patient. And what we found is by taking this approach, we've been able to reduce our manufacturing time from its current 1 week turnaround to a same-day process where we go from leukopec to cryopreserved drug product within about 10 hours. And by making the process that rapid, it dramatically drives cost of goods down, and you end up with an autologous process that has comparable costs to a truly off-the-shelf product. And by making the process quite short, you also can take advantage of things like having it in a closed system without the skilled technicians that are required to oversee the process for longer cell culturing approaches. So it's quite a unique opportunity that we have with monocytes because of the lack of need of expansion or activation to make the engineered product.
Yeah, that's great. Any other thoughts on this topic?
Sure. I believe that there's space for both. So certainly for allogeneic therapies, lymphodepletion is a big part of it. And you need the immunosuppression for the cells to persist. And so in that setting, in a transplant setting, in a bridge to transplant, in hospital academic setting, certainly there is a potential play for allo. What EXUMA is focusing on is really extending CAR T into the broader oncology market and moving to the community oncology centers. And to do that, we really feel we have to develop a CAR T for patients that don't require chemotherapy in a lymphoreplete environment with no requirements for lymphodepletion. That we feel will encourage better safety profiles. And we've really spent a lot of time in engineering the lentiviruses to make sure that we get the persistence expansion in a lymphoreplete environment. And so in that's the space where we believe autologous can be a major player, and then be on a playing field with other treatments that are more frontline and are given in the community.
Yeah,
So
I concur with that. No, it's a good answer. Are there other thoughts on this topic?
Yeah, I mean, I would like to just add, I kind of agree with Michael in terms of, I think it's attractive, obviously, from the previous question about optimizing, vein to vein time with an allogeneic product. But I think it's too early to say about the efficacy, particularly in the solid tumor space. I mean, I think one issue that also rises is the issue of persistence. Do these cells persist long enough, or do you have to give multiple infusions? What does that look like? How does that increase the cost? And I think as the autologous programs improve on process development, decreasing the vein-to-vein time, I think Daniel mentioned earlier about, the importance of having in-house manufacturing as opposed to using third-party vendors, which I also agree is critical. You have more control over the process and the ability to do process improvements early on to ultimately decrease that vein-to-vein time, but also have a closed system, as you approach larger studies and commercialization ultimately. So I think it has to go in a stepwise process and optimizing the autologous and trying to understand the complexities of allo, and they're quite different. So.
Yeah, I mean, I would completely agree with all of that, right? I think it's not an either-or scenario. Auto and allo are both most likely going to coexist. And if anything, the efficacy problems, are not solved by allo, right? It's very much more about costs and manufacturing. Bridging that gap on how to make autologous or allotherapy, just in terms of curing cancer, work better, I think is, a wholly different problem. And for us, for example, at Refuge, we're relatively agnostic. We focus on being able to have cells react in their environment so that they can, in the tumor microenvironment, as we started on the panel today, sort of be able to become more inflamed. So, for example, we can get T cells to secrete IL-12 locally, and at the same time, we'll be able to modulate CJON, which is more of a master upregulator or master regulator of things like the checkpoint genes, to be able to then handle durable responses, to be able to control persistence and things like this. And all of that is applicable in both an autologous as well as an allo context. And we would work with both types of cells whenever they're, today mostly autologous is available, of course, and allo is very much in development. But if it comes to the fore, we are very much able to leverage that. And I would say, sure, if it comes to the fore, it'd be great. But in the meantime, there's still a lot of work to be done to actually bridge that gap, making things work.
Yeah, I like all your answers, and obviously the jury is out. There is no guarantee yet where we will end up with allogeneic, and there is a place for both. But clearly, autologous is the immediate future. I think we all know that. So, let me make a transition to a different kind of question that's related to what we already spoke about. Given the complexity of the space and the significant number of innovations that we see, it's sometimes very hard to bring it all together to make a real story out of it. If integration is truly needed of different technologies for optimizing a cell therapy approach. So from that perspective of bringing technologies together, how do you as the innovators think about partnerships, either between biotechs or, of course, with the pharma partner that has a bigger ability to integrate technologies build platforms and, bring things together. So how do you feel about, partnerships in this space in a variety of shapes and forms? And I'd be happy to speak to that too. I just, let you go first.
Maybe I'll start. I think that pharma has demonstrated, even two big pharmas have demonstrated that they can partner together to bring better solutions to patients. I think where it becomes complex is that when you need products and technology from multiple partners, and that's at this point in time, it is still the case, in cell therapy that very rarely has one company, all of the pieces of the puzzle in-house. So I think, and absolutely, partnerships are critical between different biotech companies and between biotech and big pharma, but we should also try to reduce the complexity to what are viable clinical solutions.
Yeah. Other comments on this topic before I provide one?
I think I can maybe add a small snippet, which I think, it's obviously very important to the industry. Not least of all for the traditional reasons for why we do partnerships between biotech and big pharma. But I think especially with the diversity of the range of different things out there and the complexity that cell therapy brings, right? Traditionally, small molecules is a very simple concept in comparison. And that means that, Daniel, especially to your point as well, right? There's lots of things to integrate together. And as biotechs, it's very rarely possible for us in terms of whether it's financial resources or human resources to really be able to do that type of integration. And so forever, we're relatively speaking going to be fragmented. And therefore, the people who are able to do that are most definitely going to be the bigger institutions, the big cap pharma out there. And therefore, I think there is a role there where, there's a happy combination of the industry there for, bigger resource partners to be able to provide that consolidation so that there's, that ultimate, more effective cell therapy for patients and being able to collate that together.
Thank you, Jing. Any other comments on this?
So I can have a brief comment. So having recently moved over from pharma to EXUMA, it's really a very natural connection. And there's strengths in both biotech and in pharma. I think that in biotech, we're nimble and small, but the resources, the global reach, the late stage clinical development, that's really a strength of pharma, and collaboration and then really co-development I've seen a lot of success in programs that way.
Yeah, and I concur with that notion. It's actually quite similar to what we do at GSK. We have basically looked at integration of different technologies on a existing CMC platform that allows you to take one module, one piece of innovation from one partner, another piece of innovation from another partner, and bring them together to the degree that is agreeable, and then eventually think ahead and build a model under which multiple partners can come together in the form of a consortium or a conglomerate of different contributors of innovation to produce an environment in which many things can be tested in parallel or side by side, and the best technologies will be advanced. We are trying that in a small scale right now. Making that bigger will require a lot more partnerships. So I think this is the future. Cell therapy, sharing innovation, collaborating strongly will make a difference. And then eventually, partner with those that can manufacture at scale if you can't build that yourself. And we're seeing all kinds of new models, like Elevate Bio has created a possibility for that. Resilience has come on the scene recently. I'm not yet sure how far that will go, but it's definitely exciting and another opportunity. And so you will see collaborations. There have to be for us to optimize the space. And maybe Ben has a thought on this, because Ben is an observer. He's not in the middle of a company, but he sees the whole space. So Ben, what do you think about it?
I think they're generally viewed as very positive and validating. And I think that last point is what investors really focus on. If it's a new technology with new risk, just to have someone else who's viewed as an expert, throw money at something in time, that's validating. But so I think that wraps up this panel. Thank you, everyone. It was a great conversation. Much appreciated.
Thank you.
Thank you.
Thanks, everybody. Great conversation. Thank you for your time and effort.